Welcome to this session on new developments in medical AI. I'm Shreyas Vasanawala, a professor of radiology at Stanford and the radiologist-in-chief at Stanford Children's. My interests and efforts revolve around improving pediatric imaging and on developing new data-driven approaches to medical imaging. The goal of this session is to highlight the cutting-edge advances in AI that are on the horizon and that have the potential to exhibit large-scale clinical impact. In this session, you will see representative developments in the core areas of medicine. The breadth of activity and applications across medicine is quite astounding. For example, you'll see an application that benefits the smooth and skillful execution of surgeries. In a completely different sphere, you'll see a unique approach that leverages AI to repurpose existing drugs to a disease that we're all acutely interested in at the moment. Additionally, you will see an overview of applications in medical imaging, which is close to my heart. Here you will see promising ways of generating high-quality images that are either faster or with less radiation. To me, I'm highly impressed with the rapidity of the advances in the field, and I'm convinced that we're only in the early innings of this story. It's now my pleasure to introduce our first speaker, Serena Yeung. Dr. Yeung is an assistant professor of biomedical data science and by courtesy of computer science and electrical engineering at Stanford University. Her research interests are in the areas of computer vision, machine learning, and deep learning, focusing on applications to healthcare. She also serves as the Associate Director of Data Science for the AIMI Center and is affiliated with the Stanford Clinical Excellence Research Center. Thank you, Serena, for this presentation.
In my talk today, I'd like to introduce some of our group's ongoing work on developing AI to augment surgeon performance. Surgery is one of the major operations in hospitals. It has the potential to cure many diseases, but it's also a challenging environment that requires a high degree of skill. Post-operative mortality has been reported as the third leading cause of death worldwide, and studies have shown that surgical technique during operations has a significant effect on patient outcome. So one of the questions that my research group and our collaborators have been thinking about is how might we be able to use AI to augment surgeons during these critically important times during procedures? Well, video can capture a lot of information about what's happening during surgeries, and it turns out that surgeons are sometimes already capturing video data. For example, laparoscopic surgeries are video-based and already produce a video stream. And for traditional open surgeries, surgeons sometimes capture video through head-mounted or overhead-mounted cameras. And one reason that surgeons already do this is actually to review and study surgical technique. And so this highlights the value of the information that's in surgical video data. And so it naturally follows that developing AI, which can automatically recognize and interpret what's happening in surgical videos, could be highly valuable for surgeons. As an example of two major application areas, one use case could be providing assessment and feedback on surgical skill for training purposes. And another use case is providing real-time situational awareness. AI models that could interpret what's happening in the surgery and compare this with thousands of other videos on which it's been exposed to during training could be useful for providing feedback on decision-making and risks. So in our research, the first type of surgeries that we targeted were laparoscopic surgery videos. These surgeries are video-based, where surgeons insert a tiny camera and tools through small incisions into the body. And since the field of view here consists primarily of tools manipulating body tissue, we started by training a deep learning-based algorithm to perform object detection of tools in the video. And so here in the example that I was showing, you can see tools being detected in a sample video. Now, once we have the tool detections, we can produce analytics of tool usage over time to gain insight into surgeon technique. And so here I've shown 4 example videos, one in each column. And in the first row, I've shown the presence of different tools detected over time. The second row shows spatial heat maps of tool location over time. And the third row shows more detailed tool trajectories during a specific portion of the surgery. If we compute some statistics based on the detections, we can see here on the left the total time that each instrument is used in each of the videos, and on the right, the total distance traveled by the instruments. These statistics can provide some interesting information. For example, video 3 here has higher usage of the bipolar instrument, which is used to stop bleeding, and so this could be an indication of tissue damage. On the other hand, on the right, we can see that video 2 is the most economical in terms of motion, which is a characteristic that's usually correlated with higher skill. For these videos, we also had 3 surgeons manually watch and rate each of the videos using the GOLS ratings for laparoscopic surgery, which is an established scale for laparoscopic surgery skill. Here, higher scores are better, out of a range from 1 to 5 for each category, and we can see that the surgeons overall rated video 2 to have the highest skill and video 3 to have the lowest skill, which is consistent with some of the insight that we gleaned earlier. While this was still a small study, I think it demonstrates the promise of AI-based surgical video analysis, and we're now scaling this work to a larger scale. Now, laparoscopic surgery is great because it's already video-based, but open surgery still comprise the majority of surgeries. We can imagine that AI is likely very useful for analyzing open surgery videos as well. However, this setting is significantly more challenging. First of all, open surgery videos can be collected in much more diverse settings with different viewpoints such as head-worn or overhead-mounted cameras, as well as with larger fields of view showing surgeon hands, anatomy, and other elements of the surgery scene. Most existing work on analyzing open surgery so far have therefore relied on non-visual sensors, such as motion tracking sensors. And finally, since data capture is much less standardized than for laparoscopic surgeries, there currently lacks data sets to support the development of AI algorithms. So faced with these challenges, we wondered if we could pull videos from many different sources in a crowdsourced manner. We looked at one of the largest crowdsourced repositories of all kinds of videos, which is YouTube, and thought, how often might surgeons be posting videos of surgeries on YouTube? Well, it turns out that it's actually quite often, and YouTube is a very rich and active repository of surgery videos. And so in our work that was led by our group members shown here, Emmett, Krishna, Michael, Stephen, and Rohan, as well as our collaborators at Harvard and Beth Israel Deaconess Medical Center, led by Dr. Gabriel Bratt, we proceeded with a methodical scraping of thousands of surgery videos under several major categories: breast, gastrointestinal, and head and neck surgeries. Here you can see a word cloud showing some of the popular types of surgeries that appear in our data set. Now, once we obtained this data set, we turned to developing computer vision algorithms to recognize various aspects of the surgical scene, and we annotated the data set to support this algorithm development. Specifically, we targeted the temporal detection of several common surgical actions: cutting, tying, and suturing. And we also targeted detecting hand locations and hand poses based on the key points shown here. And finally, we targeted detecting several major tools: the Bovie, needle driver, and forceps. To obtain an AI algorithm that could take a video as input and simultaneously produce a rich set of outputs, both effectively and efficiently, we developed a multitask deep learning model. Our model is comprised of a shared backbone of neural network layers to extract useful lower-level features, as well as task-specific heads that reason on top of these features to output action, hand, and tool detections. In the end, we were able to train a model that could perform simultaneous recognition of actions, hands, and tools in the surgical scene at real-time speed. Here's an example showing an early version of our model in action, where you can see the detected hands and hand key points inside the green boxes, tools in the blue boxes, and action detections in the moving bar at the bottom. Now, with this model, we're again able to obtain detailed characterizations of videos. For example, we can plot the sequence of actions occurring in a video. I've shown this here for 30 appendectomy videos, and we can start to see broad patterns occurring, with lots of cutting in the first part of the surgeries, followed by suturing and tying later on. And we can also average these to obtain the prototypical structure of an appendectomy shown here on the right. Finally, as we did with the tools in laparoscopic surgery, we can also analyze movement. Here I'm showing the movements of hands over a period of time overlaid on the surgical video, where we've used blue to indicate slower and steadier motion and red to indicate faster motion. And in this example, you can see that the surgeon on the left has an overall steadier trajectory, which is a characteristic of higher skill, while the surgeon on the right has a wide distribution of velocities, including larger jerks, which is a characteristic of lower skill. So I hope this illustrates some of the interpretation and analysis of surgical videos that's possible using AI. Finally, one other key component that we'd like in AI analysis of surgical videos that I haven't mentioned yet is recognition and in particular segmentation of anatomical structures. As we work towards applications for real-time situational awareness, Anatomy will be critical for situating actions and providing context-aware assistance, such as warning that you may be at risk for injuring a particular anatomy. In a project that's led by my student Julia Gong and our collaborator Chris Holzinger at Stanford Head and Neck Surgery, we've trained computer vision models to be able to segment an important tissue, the recurrent laryngeal nerve, in thyroidectomy surgeries. This is a critical soft tissue that's important not to damage, and so we want to be able to identify it. To approach this problem, we first collected a data set of digital camera images containing the nerve from diverse conditions, including different lighting conditions and different levels of zoom. We then developed a segmentation model that first performs automated detection and pre-processing of the wound region to obtain standardized images that are robust to image capture conditions, and then we trained a masked RCNN model to predict the output mask of the nerve. In addition to this, one really useful aspect of AI-based interpretation is that it can also provide richer interpretations, such as not just recognizing the nerve, but also producing automated measurements of the nerve. In this case, since the retractor tool in the surgeries has a standard known size, we can use AI to detect both the nerve as well as the retractor tool and calibrate their relative widths to estimate the width of the nerve. Finally, here are some examples of estimated nerve width based on the predicted nerve segmentations, boxed in red, and the predicted retractor segmentations, boxed in green. So to conclude, today I've shown some of our ongoing work on developing computer vision to be able to recognize and interpret what's happening in the operative field during surgical procedures using surgical video and image data. And while we're still in the early stages of research in this domain, I believe that as we develop computer vision to extract richer and richer information from this data, that we'll be able to use it to develop valuable ways of augmenting surgeons in both training as well as during the operations themselves. Finally, I'd like to acknowledge the members of my research group, and thank you.
Thank you very much, Dr. Yeung, for that wonderful presentation. And now we'll hear from Pranav Rajpurkar. Pranav is an assistant professor at Harvard University in the Department of Biomedical Informatics. His research approaches problems in clinical medicine with a computational lens, developing algorithms and data sets that can drive AI technologies to support medical decision-making. He co-hosts the AI Health podcast and co-edits the Dr. Penguin AI Health newsletter. He instructed the Coursera course series on AI for medicine and founded the AI for Healthcare Bootcamp program. Previously, he completed his PhD program, co-advised by Dr. Andrew Ng and Percy Liang at Stanford. Pranav, thank you very much for giving this talk.
Hi, I thought I'd chat today about what I consider to be one of the most important problems facing the field of medical AI, which is the reliance on time-consuming manual annotation by medical experts. Traditionally, the successful development of deep learning algorithms for medicine and beyond medicine has relied on the availability of large annotated data sets. And one of the advances that's promising to be able to overcome this challenge is self-supervised learning. Self-supervised approaches have recently demonstrated many successful applications, being able to learn representations without labels to produce pre-trained models for subsequent fine-tuning on labeled data. The most competitive methods for learning representations without labels have been self-supervised contrastive learning. These methods learn representations through a contrastive loss, which pushes apart dissimilar data pairs while pulling together similar data pairs. I want to dive into some recent work that I led demonstrating the successful adaptation of contrastive learning from the natural image setting to the medical image setting, and then also talk about the development of methods that take into account medical metadata. So let's dive in. As background, in contrastive learning, the selection of pairs controls the information contained in the learned representations. For natural images, positive pairs are typically created using different augmented views of the same image. On the other hand, negative pairs are views of different images. So to formalize this, given a set of augmentations T and an encoder G. Most contrastive learning algorithms involve minimizing the info NCE loss. Here, for a query image X, we define two transformations, X1 from transformation T1 of X, and augmentation X2 from transformation T2 of X. The negative pairs for X1 are the transformations of other images, ZI, and these ZIs come either from a Q in the case of a method like momentum contrast, or the mini-batch in the case of a method like SimClear. So I led a study late last year demonstrating that we can make a straightforward modification to the data augmentation strategy by defining the set of transformations T to be a composition of augmentations commonly used for chest X-ray interpretation. And using this relatively straightforward modification, we found that using just a small fraction of labeled data, a model pre-trained using contrastive learning could have a huge boost in performance on a data set, especially only when a fraction of that data is labeled. To the best of our knowledge, this work was the first to show the application of contrastive learning to chest X-ray interpretation. A team at Google has since demonstrated a similar technique on chest X-ray classification as well as on dermatology, skin condition classification, and at the NYU School of Medicine, a similar method is applied in their recent work in collaboration with Facebook for using chest X-rays for COVID-19 risk prediction. Now, despite the early successes of these applications, these contrastive learning studies on medical imaging have not systematically investigated how to leverage patient metadata available in medical imaging data sets. And in this recent study, we looked at how we can investigate more formally how to select these positive pairs using patient metadata. The key idea in this work was that we can use metadata associated with a query image X, such as the patient number or the study number or the laterality, to create positive pairs. We can use this patient metadata to obtain an enhanced augmentation set, which is dependent on X, where we first collect images from the same patient P, and then create a set SC of X, which is the set of all images satisfying some predefined criteria C in relation to the properties of X. We can then sample from this enhanced augmentation set such that when it's non-empty, otherwise we just default to using an augmentation of the same image, and this definition just lays that out more formally. The key in our formulation is that it provides us the flexibility of experimenting with what is this criteria C constraining the set SC using the metadata for the query X. In particular, we focus on two properties. One is the study number of an image associated with a particular patient that reflects the session or the time point that the image was taken. So images from two different studies, which may be separated by years, while images in the same study are acquired at the same time. And we experimented with only using images from the same study and from different studies. Similarly, the second property we looked at was laterality, as chest X-rays can either be frontal, which is AP or PA, or lateral. And we experiment with only keeping images from the same or different laterality, or keeping it unrestricted. And our results clearly show improvements with this incorporation of metadata. We see some large improvement from the baseline, with the best result obtained when using the set of images from the same patient and same study as that of X, regardless of the laterality. We perform experiments to test hypotheses associated with these differences in performance. For example, we see that there is a drop in performance when moving from using images with the same study number to using images without regard for study number. We hypothesize this is because all studies may contain images of a different pathology than seen in X. As a result, the model is asked to push the representations of a diseased image close to the representation of a non-diseased image, causing poor downstream performance. To test this hypothesis, we carry out an oracle experiment where we constrain the positive pairs to only include images from studies that have the same downstream label as that of X. And we find that the model pre-trained with this constraint achieves a higher performance than without this constraint. And this experiment supports our hypothesis that positive pairs from images with different downstream labels hurt performance. Similarly, we also investigate the performance drop when moving from using images across all studies of the same patient to images with a different study number from the current query image. This finding may also support our hypothesis that positive pairs from images with different downstream labels hurts performance because there is a larger proportion of positive pairs of different disease pathologies in pairs of images from strictly different studies. Here we have to make sure we are controlling for the size of the available sets and that we're not using any contributions from positive pairs formed from the same image. So we implement these two controls. Here we see that the all studies strategy outperforms the same patient distinct study strategy, even when the size of the augmentation set is controlled. A controlled experiments thus show that one of the keys to improving downstream performance on disease classification is to use patient metadata to appropriately create positive pairs from different images with the same underlying pathologies. So I think self-supervised learning, in summary, will have huge applications in medicine, especially since it's defined by the presence of large quantities of unlabeled data. And one particularly interesting opportunity is that we can use heterogeneous data sources to be able to set up these self-supervised tasks and make fast progress. And I'm interested in collaborating further with folks to investigate how we can get models not to rely so much on labeled data, which has been a problem in the field for a while, but self-supervised learning promises a strategy to perhaps get around this very successfully. I'll stop there.
Thank you, Pranav, for that wonderful talk. And now we'll hear from Marinka Zitnik. Dr. Zitnik is an assistant professor at Harvard University with appointments in the Department of Biomedical Informatics, the Broad Institute of MIT and Harvard, and Harvard Data Science. Dr. Zitnik has published extensively in top AI/ML venues and leading journals. Her algorithms are used by major institutions including Baylor College of Medicine, Karolinska Institute, Stanford Medical School, Massachusetts General Hospital, and the pharmaceutical industry. Thank you, Marinka, for giving this talk.
Today I will talk about actionable AI for drug discovery and development. Drug discovery and development is an incredibly resource-intensive and expensive process. In fact, it takes between 12 to 16 years on average to develop a new drug from scratch, and it can cost $1 to $2 billion. And there are multiple reasons why this process is so challenging. That relates to high failure rate of drugs, as well as a variety of challenges that exist in each of the stages of the development phase that need to be successful in order for the candidate compound to advance to the next stage. So faced with a skyrocketing cost of drugs, in times where rapid therapeutic innovation is needed, this traditional iterative development and experimental and clinical testing and approval of new drugs is really not feasible. In COVID-19 pandemic, we were faced with rapid development and deployment of safe and effective therapeutics was needed. And so enticing alternative to developing a drug from scratch is to repurpose an existing drug in order to identify what drugs are already on the market that might likely have therapeutic effect in COVID-19 patients. So in essence, what we are trying to do is to find new tricks for old drugs, for existing drugs. And so the project that I will highlight in this talk today primarily took place between March and June 15 last year, where with a large team of collaborators, we were able to compress months and in some cases even years of work into weeks and days through the use of automation and artificial intelligence algorithms that we have been developing. And that led to an end-to-end pipeline that allowed us to bring and collect a variety of heterogeneous data, produce ranked lists of predicted drugs that represent candidate therapeutic opportunities for COVID-19, and then validate experimentally most promising to the lab. So the core project is what drug treats what disease. So that is the fundamental test that we would like our machine learning models being able to answer. And so that is challenging because the way drugs work is not only by affecting only the proteins to which they directly bind, and instead the effects of drugs propagate through the underlying biological networks that exist at multiple scales of biological complexity. So because of that, it is important to be able to consider a very heterogeneous, rich and interconnected data at multiple scales in order to predict what diseases a given compound might treat. So in our case, the first question to answer when trying to pursue this endeavor of computational drug repurposing was to think about how to computationally represent COVID-19, how to make represent the data in such a way that this is amenable to machine learning analysis in the context of rich interconnected biological networks. To this end, we relied on data produced by Gordon et al. From UCSF, who have expressed 26 out of 29 SARS-CoV-2 proteins, and they use mass spectrometry to identify 332 human proteins to which viral proteins bind. And so this set of 332 human proteins and the way those proteins interact with each other in human cells is what we use to define COVID-19 disease module, a set of human proteins that are directly attacked by the virus. And we overlaid that information on the viral disease module on top of the human protein interaction network, which is a network of where nodes represent human proteins and edges indicate contacts between those proteins. The question then becomes whether proteins in immediate vicinity of the viral disease modules tend to be proteins that are targeted by some of the existing drugs, and whether those drugs would be effective treatments for COVID-19. To scale up that idea, we constructed a large repurposing knowledge graph that consisted not only of information of the human protein interaction network and the COVID-19 disease module, but also had information for all approved drugs in the US, and for each drug, what are the proteins to which that drug binds. And for each of those drugs, a drug subgraph or drug module that was overlaid on top of the protein interaction graph. In addition to that, we also had a large number, a few tens of thousands of different disease modules for common diseases for which treatments already exist. And each disease was represented by a module of proteins associated with the onset of that disease. With that data in place, the goal was, given common diseases and existing treatments for them, can we predict candidate treatments for COVID-19? And from the machine learning standpoint, this is a challenging problem because it requires us to reason about a never before seen disease. In March 2020, the COVID-19 disease was associated with very little available data. In particular, no labeled examples of successful treatments. And so that required novel algorithmic innovation, in particular, the development of new few-shot learning algorithms for graphs, which we describe the details for in the paper cited on this slide. When using the few-shot learning algorithms and applying them to our graphs, the first experiment that we did was to test whether those drugs that were predicted by the algorithms as most promising effective therapeutics tend to be enriched for drugs that were tested in clinical trials at that point in time. So we used information on clinical trials as surrogate values to estimate performance of the methods. And what I'm showing on the left part of the slide is the AUROC curve that compares our graph neural network-based few-shot learning methods against a variety of alternative network science methods. And overall, the results were incredibly promising, and that has led us to taking predictions produced by these methods in order to generate a final ranked list of drugs that were then shared with our experimental collaborators. Collaborators from national emerging infectious disease laboratories have took predictions produced by the algorithm and screened them in human cells. And so the result of that process was that top-ranked drugs, as returned by the algorithm, yield a 62% success rate in the sense that they reduce viral growth, which is in contrast to 0.8% hit rate that would be obtained by non-guided experimental screening without the use of our algorithms. And so that is significant because it's an order of magnitude higher hit rate than what alternative approaches would allow. By engaging in this very intensive collaboration and close interactions and work with experimental biologists, virologists, and biomedical researchers, there are a number of takeaway messages that we learned and insights that we gained that can inform future algorithmic development. In particular, one such insight that I would like to highlight is the strong need for actionable predictions. Those are predictions that are not only accurate, but that allow scientists to build trust into predictions produced by the model, that allow scientists to interpret them in a meaningful way and then provide feedback that can then engage in model development. So we were lucky that we were able to deploy some of the algorithms on explainable AI for graphs that we have previously developed and use them for this project. And this process has resulted in a discovery of a new class of drugs that we call network drugs. So in particular, what we did is we looked at what are drugs that were correctly predicted and experimentally verified, and what is common to those drugs. 76 out of 77 drugs that successfully reduced viral infection turned out to be drugs that do not directly bind to proteins that are targeted by SARS-CoV-2 virus. Instead, those drugs rely on network-based actions and cannot be identified by alternative docking-based strategies. And we named that class of drugs as network-based drugs because the way they operate and exert their therapeutic effect is by binding to proteins that in turn interact with some other proteins that eventually interact with proteins that are attacked by the virus itself. So the project that I highlighted gives an example and provides a vignette into really tremendous and incredible opportunities and for innovation and impact that drug discovery and development offers for machine learning community. But to be really able to operationalize and realize those opportunities to the fullest extent, to have AI-ready data sets and meaningful tasks for therapeutics. To this end, we founded an initiative that is called Therapeutics Data Commons, available on the website tdcommons.ai, which is an open science initiative of machine learning-ready data sets, as well as an ecosystem of tools and molecule generation oracles, strategies for performance evaluation that can facilitate algorithmic and scientific advance in the broad area of therapeutics. And so Therapeutics Data Commons is the first initiative of its kind, and I highly encourage everyone to check it out as well as to contribute to this growing community of therapeutics machine learning. With that, I will conclude. Papers, code, and data that I have mentioned today are all available at our lab website. And
Thank you, Marinka, for that wonderful presentation. And now we'll hear from Dr. Akshay Chaudhari, who's an assistant professor of radiology in the Integrative Biomedical Imaging Informatics program at Stanford. He's interested in the application of artificial intelligence techniques to all aspects of medical imaging, including automated scheduling and reading prioritization, image reconstruction, quantitative analysis, and the prediction of patient outcomes.
Good afternoon. Thank you very much for the kind invitation to speak at this year's AIMI Symposium. I'm delighted to be here to share some of my thoughts on how AI can be used for different use cases beyond image interpretation and can actually be beneficial for generating the medical images that we work with on a day in and day out basis. So as we begin, I wanted to start off with a very high level perspective of how the diagnostic radiology process works. The first step for any medical imaging modality is the acquisition of some raw data. Then, once we've acquired this raw data, we can use our knowledge of the underlying physics of the system to generate the medical images that we want to be able to image. And then once these images are generated, we can either have these undergo radiologist review, or we can build automated algorithms for automated analysis of these images. Typically, anything that comes before the image generation phase, the acquisition and the reconstruction parts, these methods that involve AI are typically termed as upstream AI processes, while everything that comes after the image generation phase is typically referred to as downstream AI applications. And in this symposium, we've heard a lot of use cases for downstream AI. But here in this talk, I wanted to provide a few examples of how different upstream AI processes can actually benefit diagnostic radiology across different modalities. For example, for computed tomography, we can acquire low radiation dose images and using deep learning, enhance the image quality to improve the diagnostic utility that those images can have. For MRI, we can undersample our data and go from artifactual images to nice images that have diagnostic quality also. In the case of PET imaging, we can acquire these PET images with either a low scan duration time or with reduced radiotracer dosage and still be able to use deep learning to improve the image quality. In ultrasound, we can perform unsupervised despeckling of the images to improve the image quality for downstream interpretation. So there's a lot of potential use cases that upstream AI can have in radiology, but for the short talk today, the focus of my presentation will really be focusing on how we can use some of these upstream AI models to enhance the value of MRI for accelerated imaging. And I think MRI is particularly ripe to innovations because most clinical protocols can take anywhere of 20 to 30 to 40 minutes of scan time. So reducing that scan time has the potential to increase patient comfort. It can decrease healthcare costs, and it may provide the ability to also add new, more sensitive sequences for looking at early biological changes. So very quickly, let's cover how MR works to understand different acceleration processes downstream. Say I wanted to image this axial knee MR scan, and this is the image I want to generate. Well, in an MR scanner, the data that I'll be sampling is actually the Fourier transform of this knee. And we refer to this as the K-space. Once we have this K-space, we can simply perform an inverse Fourier transform to generate the image that we've wanted to acquire. However, the downside of this process is that sampling all this K-space requires a fair amount of scan time. So in order to reduce that scan time, we can think of different ways in which we can undersample this raw data. And now once we have this limited undersampled K-space data set, if we naively perform an inverse Fourier transform, most of the radiologists in attendance will probably be pretty sad looking at this low quality image. So fundamentally, the goal of most MR reconstruction projects is to be able to balance what is the least amount of data that I can sample while still generating images that have high diagnostic quality. So towards this end, some of the first work that we did was exploring computer vision-based super resolution in order to convert low resolution MR scans to high resolution MR scans. And this was to replace the traditional interpolation algorithms that are used either directly on the MR scanner or in PACS viewers, such as bicubic or tricubic interpolation. So let's say we started off with some blurry input image. Here we're looking at a coronal scan of the knee, where some tissues such as the medial collateral ligament are considerably blurred out. However, once we pass this through our super resolution algorithm, we can see that this medial collateral ligament and other sharp tissues have much better image appearance than the low resolution input images. And we can actually compare this to some ground truth acquisitions that took twice as long to acquire, and we see very high concordance between the ground truth and these super resolved images. Furthermore, we're able to use some of these super resolution images to still enable accurate tissue segmentation of fine structures such as the cartilage and the meniscus, despite acquiring low resolution scans in the first place. In addition, we're also able to show that super resolution can enable quantitative MRI, where we can actually go beyond simply imaging the morphology of tissues, but also be able to look into the biochemical structure and be able to quantify early degenerative changes in collagenous tissues. In addition, we're also able to prospectively deploy the sequence in the clinic, and we showed that a single 5-minute knee MRI scan can provide the same diagnostic information as a traditional 25-minute knee MRI protocol. And we showed this in a cohort of 50 consecutive patients. So while there's a lot of benefits of these super resolution approaches, we wanted to still go back to the drawing table because we still know a lot about the MR reconstruction process over the last 3 or 4 decades of knowledge. So our next question was, well, can we actually take what we know about MR physics and induct that into some of the models that we're building? So in order to do this, we can still start off with our undersampled case space as we do. We can come up with an initial reconstruction, but now we can actually inject some knowledge of the underlying physics. So about some of the coil sensitivity maps, as well as some of the MR relaxation parameters. And now these are all the information that are being fed into a neural network. Here, we build an unrolled neural network that consists of multiple repeating blocks. One block is a CNN block, which, as I showed you before, can improve the image quality of our input. But at the same time, there's also this data consistency blocks, which make sure that whatever the CNN is adding into the image is actually consistent with the actual acquired data. In this way, we minimize the chance of any hallucinations or adding features that shouldn't be there in the images. And we can implement these DC and CNN blocks in a cascade to mimic proximal conjugate gradient descent and generate the output images that we care about. So using this physics-driven deep learning approaches, we can actually use a lot of our domain knowledge to reduce how ill-posed these problems are. And using this, we can see that even with 8-fold undersampling, we can build 3D deep learning-based reconstruction algorithms with image quality that's near identical to these reference scans. So despite these images still being that good, we still wanted to push this method a little bit further. And the next question that we wanted to ask ourselves is, well, can we move away from this notion of fully supervised training? And maybe the most extreme example of that is, what if we have no training data at all? Can we still be in this scenario where we can use image reconstruction networks to improve MR image quality? And I think the answer is yes, based on some fundamental insights about neural networks themselves, because they can fit the signal better than they can fit the noise. And let's look at a graphical example for this. Say I start off with some corrupted image. This image has gone through extreme JPEG compression, and we see some artifacts. There's black speckles here. There's these blotchy artifacts that can be seen next to these sharp transitions. And all we ask a neural network to do is recreate this image. That's it. So during the early stages of that neural network training, we can see that that network is building some semblance of an image. It's getting its colors right and then getting the general shapes. Over time, it's converging to a better output. And then eventually we actually get to a space where the output actually is much sharper than the input. And the reason this happens is because the network is actually not capable of fitting to the random noise and the random artifacts. And because of that, it ends up acting as an implicit super resolution network that can also denoise simultaneously. So using these insights, we're actually able to perform unsupervised reconstruction without having any access to ground truth data. And we can see here in the case of 4-fold accelerated knee MRI, our ground truth data is on the left. Our COM decoder method, which is unsupervised, did not have access to the ground truth, but it can still reconstruct images with high image quality. and it is nearly comparable to a fully supervised reconstruction also. Furthermore, we're also able to quantify the impact of distribution shifts. So say I trained a model on NEMR scans and ran inference on the brain, and vice versa. I trained on brains and tested on knees. Well, with these unsupervised methods, it doesn't necessarily matter because there is no implicit bias, and we compute each image one by one. However, even with our data-driven deep learning-based methods that do have a physics-based injection in them, as well as a fully data-driven injection, we see that there's still certain artifacts that are propping up in the image. And this is comparable to some of the traditional acceleration methods, such as compressed sensing. So I think even though a lot of these methods can be used to accelerate images, we should always be aware of distribution shifts. And actually, we're trying to come up with different ways in which we can combine our data-driven methods that can produce high image quality, along with some of our unsupervised methods to make sure that we are not susceptible to these distribution shifts. So as a conclusion, hopefully I've shown you today that AI can be used for some upstream applications to improve the efficiency of medical image acquisition, and that really understanding the structure of our CNNs and leveraging the knowledge of our physics, hope we can actually build models that reduce how much data we need and still have accurate and robust reconstructions. And lastly, before deploying, it's important to be able to characterize the robustness and its impact on distribution shifts and be able to mitigate those if they exist. With this, I'd like to acknowledge all the students who contributed to these slides, our collaborators, and our funding sources. And I'd be happy to take any questions during the upcoming panel session. Thank you.
Well, that was a wonderful talk, Akshay. Thanks so much. And thanks again to Serena, Pranav, and Marinka. Together, your talk showed a range of very practical and innovative initiatives. We now have about 14 minutes or so for questions. If there's anybody from the audience who would like to submit one, please do so through Slido. I'll just get started with the first one, and I'll ask Serena. Serena, I find a number of websites that give assessments of physician performance, and they're rather limited. Can you envision some automated metrics of surgeon performance that are placed in a very public way that would provide a consumer some helpful advice on who to choose as a surgeon?
Yeah. So, I think the goal of a lot of what we're doing is to try and develop tools to help, in this case, surgeons to be able to continuously, improve their technique over time, whether it's during the training stage, when they're residents and so on, or whether it's, getting kind of colleague type of feedback when they're more expert surgeons. So I think that a lot of this is, I think, designed for sort of, personal development and assistance. But certainly, I think they could be used in more broader context as well. I think that here, there's a lot of discussions around this sort of area. Does it make sense for these kinds of tools to be used for personal development, for training, for certification, for others? And to be honest, I think that we don't have an answer yet on what's the best way to handle these sorts of things to make it something that's broadly useful, but is also respective of, for example, individual privacy. And so I think that's still an open question to be addressed as a field.
Wonderful, thank you. And Marinka, if I may ask you a couple of questions. I really enjoyed your presentation on repurposing drugs for COVID-19, which is quite timely. I was wondering if you had to pick the next disease to work on, which one is ripe for this type of technology?
So that's a great question. So we currently are working on several disease areas using the methodology that I described. And so one particular that we are very interested in is ALS. And for ALS, for which no good treatments exist, we are really using the power of graph neural networks and reasoning over knowledge graphs to find and prioritize higher order combinations of drugs that could be, when taken together, lead to certain beneficial effects for patients with ALS. So that's one of the focus areas now. And the other focus area goes to rare diseases, where we work with the Undiagnosed Diseases Network, or UDN, which is a nationwide initiative on diagnosing and then identifying treatments for patients with rare diseases. And that's particularly challenging because there is little incentive from some of the large pharma companies to develop treatments and therapies for patients with rare diseases, since there are typically very few of those patients. However, there are many rare diseases. So altogether, those diseases become common diseases. So we are working with that nationwide initiative in order to parse through all the data that we have on rare diseases and identified possible therapeutic opportunities using drugs that are already approved or in late clinical trials that could have a certain desired and positive therapeutic effect for those patients.
Wonderful. And if I understood your methodology correctly, it was in some ways a little bit different from the approaches that Akshay was showing, where he brought in some of the physics into the image reconstruction problem. And I wonder, in your case, is there a role for, say, quantum mechanics, or looking at the physics of interactions, that it could be integrated? And if so, how would you go about doing that, or are people doing that?
Absolutely, and there are actually people doing that. For example, with AlphaFold, that is just one of an example where models of proteins are being considered to identify what are those proteins that have the structure that would make them candidate target proteins. So there certainly is quite a bit of opportunity to develop either physics-informed or physics-preserving models that would bring in deeper insights in models and incentives and rationales, actually, from physics and chemistry, and encode them into models to improve inductive bias for models that we currently have. Our models so far primarily focus on integrating biomedical knowledge and use that information as a way to prime predictive models. But I think there is plenty of opportunity to bring in additional knowledge from chemistry and physics in order to identify what are proteins that are possible drug targets, how those proteins that in humans interact with proteins that are attacked by other infectious diseases, for example.
Great, thank you. The next question is for Akshay. Akshay, a lot of the focus of your talk was on image acquisition and reconstruction algorithms, which are all essentially software problems. Is there an interplay between this that may impact on hardware design for imaging equipment? And if so, are there some potential benefits of that?
Yeah, that's a really good question, Shreyas. And I think probably the biggest challenge that we find right now in these MR reconstruction problems is, what is the best data to acquire? If I have some raw data space that I have to physically traverse through, right now, we always think of the hardware systems being something that's fixed. And maybe this even goes back to one of the points that Andrew brought up earlier in his keynote talk about, can we actually be data-centric? If we're not constrained to the types of acquisitions we've always been used to, I think there could actually be a lot of potential for building better gradients. So we, instead of traversing our case space through Cartesian trajectories, maybe we can start using some exotic trajectories like radial or 3D cone acquisitions. But I think before we get there, it's really important to understand what are some of the potential impacts that these methods have, what are some of the robustness criteria. And I think all of these questions go hand in hand, but I absolutely think that once we get a better understanding of how far can we push our software-based reconstruction methods, I think that will definitely inform what is the best hardware that we should develop so that both of these technologies can really go hand in hand.
Great, wonderful. So the next question I have is for Pranav, and then I'd like to after that, open it up to everybody who's part of this panel. In a lot of these applications that we've seen here, there's a lot of domain-specific expertise that's required. And, in my area around imaging algorithms, that shows up in one way. I saw it also in Marinka's talk with some detailed knowledge about, biological networks. And so I wonder if somebody's thinking about getting into these fields, how do you prioritize what's important to learn and get good at? Do you really want to sort of become the subject matter expert first and then learn about some of the deep learning techniques and other machine learning approaches? Or do you come at it from sort of the machine learning part first, and then try to find an area and dive deeply in terms of making yourself an expert in that area? Pranav, if you were new coming into this, how would you go about it?
I think the sky's the limit here in terms of opportunities. One of the nice things that we've been able to do as a community over the last few years is for people coming in with backgrounds that are traditionally computer science, we've established programs and ways of having people with that background come in and learn about all of these cool things that are being done. So much so that I would argue that for someone who's trained in, let's say, computer science with 1 or 2 classes could catch up to the forefront of research in a pretty short amount of time. And that's crazy if we think about just how fast the field is moving and how much people are able to contribute in a short amount of time. On the other side, there have been plenty of people who have come in with a background in medicine or biology and have picked up the computational chops to be able to contribute to research in a meaningful way. And for folks who don't have either of these backgrounds, it's also possible to make an entryway into this field. One of my personal learnings has been to get to a stage in which I know what I don't know, in order to be able to know how I should collaborate with people who have the skills on either the domain side or the toolkit side. And I think that's one of the wonderful things about being able to find people with other expertise and work together. So I would say, just in summary, just a lot of opportunities with the educational resources available for anyone with any background to catch up and contribute to the space.
Thanks. Does anybody else have some thoughts on this question?
I guess maybe one thing that I can add is that one thing I found in my own experience is that when you start working in a new domain, let's say you're a computer scientist interfacing with the clinical domain, it can feel a little bit overwhelming that, how can I learn, let's say, everything about this particular disease area or this specialty. But I think that in terms of acquiring the knowledge that you need for your specific research, you can actually pick up quite a fair amount of maybe, relatively targeted knowledge for your specific problem. I think just by spending that time with collaborators, reading, learning, and that it's much less overwhelming than, say, thinking that you have to be an expert in the whole broader area. And so I think that's something that sometimes I think people may feel intimidated at the beginning of trying to conquer so much, but that if you kind of start with your specific research area, I think, and just go for it, I think you can actually learn quite a bit.
I'd like to echo what Serena said and really try to bring out, I think, at least in my career, the role that clinical collaborators have played. And I always try to think of me solving engineering problems for clinical needs. So without having a good clinical collaborator on board, I may not even know what the real clinical need is. I may think that something else is a problem when in reality, it really isn't. So I feel like, especially someone coming in with a technical background, really engaging with the clinicians, really trying to understand their workflows can be quite helpful. Just as an example that comes to mind, when I was developing some new image acquisition methods for MRI, I spent a month or so in the MSK reading rooms, just walking behind radiologist shoulders, trying to peek what they're doing, trying to talk to them. And I found that immensely helpful just to understand what the real bottlenecks were, so that when I go into the lab, hopefully I'll be working on problems that are actual problems in the clinic.
Yeah, I would certainly second that. And for example, this symposium that we are at today, it's a wonderful example of the kind of meeting points that we need that would bring together AI scientists and AI specialists with the domain scientists in order to all create opportunities for teaming and collaboration. Because what I see as most significant problem is really making sure that we work on problems that are impactful and are really open challenges in a particular area of biology or medicine. Otherwise, it can end up happening that we spent quite a bit of effort on developing algorithms that are not necessarily addressing the problem that is challenging. And so because of that, it's really important. And I very much appreciate this kind of events being organized that can bring together diverse audiences.
Wonderful. Well, thank you so much to each of the panelists. Our time for the discussion went very quickly. And I want to thank again everybody who's in the audience for listening to the session. There are a couple of questions that are in Slido. So if the presenters have a chance to take a look there, please do, and maybe they can be answered right there. So thank you again to everyone.